Today's chapter is biotechnology and its application. Biotechnology, as you studied in the last chapter, biotechnology principles you have covered already. So in this chapter we will deal what are the different applications we can have with the help of biotechnology, especially with recombinant DNA technology. So basically there are three criteria in the research area of biotechnology. First is providing the best catalyst in the form of improved organism, usually a microbe or a pure enzyme. Second criteria would be creating optimal conditions through engineering for a catalyst to act. So this is related to what we have done in recombinant DNA technology, that providing the product. What is that product? Protein product. इसके साथ ये होता है, चलना बंद हो जाता है. So through recombinant DNA technology, we have inserted the desired gene in the host organism, and from that we obtain the recombinant protein or the desired protein that can have the catalytic property also, or whatever use we have acquired from the microbes in the form of enzyme or in the form of hormone or proteins. Third thing which is required in biotechnological research is downstreaming processes. Downstream process, I already explained you. Downstreaming is when we have any protein, desired protein, through recombinant DNA technology, that should be purified and then it is separated from the rest of the chemicals whichever we have extracted, and then it is refined into a pure form, that is downstream processing. Also for stabilization some preservatives are also added. So that process is downstream processing, which purifies the protein and organic compound which you have extracted through DNA, recombinant DNA technology. So there are various applications of biotechnology, but in this chapter we will cover two basic applications. First is in the field of agriculture and second in the field of medicines. Then we also study about the transgenic animals and some plants also, and what are the issues related, ethical issues related to the use or exploitation of the biotechnology in the field of agriculture and animal science. So first topic of today is biotechnological applications in agriculture. So let us look at the three options that can be thought for increasing food production. Agriculture, as we all know, agriculture is related to producing food for humans and animals too. So to obtain food production, three requirements are there. First is agrochemical based agriculture, third organic agriculture, and second organic agriculture, and third genetically engineered crop based agriculture. Now the first, agrochemical based agriculture and organic agriculture, these are two traditional forms of agriculture which are in operation since long period of time, as we have known the agriculture. Now this third technology, which is genetically engineered crop based agriculture, deals with the technology we have studied in the last chapter, that is biotechnology, which is including the utilization of or you can say manipulation of the genetic base of the desired organism. So before going into detail about what is genetically engineered crop based agriculture, first we have to see the agriculture, how it has been started. So green revolution is there in the field of agriculture. The first revolution, that is green revolution, which had aim of increasing the food supply, increasing the food crop production to fulfill the demands of the country requirement. So green revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Hence green revolution is helpful, but the population growth is much more as compared to the production increase in the production. Therefore, increased yields have partly been due to the use of improved crop variety. So there are some reasons which has uh, supported in the increment of the food. First is use of improved crop varieties, but mainly due to the use of better management practice and use of agrochemicals, that is fertilizer and pesticides. So not only improved varieties but also utilization of agrochemicals like chemical fertilizers and pesticide are helped in increasing the yield. However, for farmers in developing world, agrochemicals are often too expensive. Yes, it is correct. Agrochemicals are expensive. They are synthesized in the industries at a larger scale. Further increase in the yield with existing variety is not possible. That is another difficulty in today's scenario. Without the use of agrochemical and utilizing the existing varieties, whatever varieties we have right now, we cannot increase the production. There are the reasons behind it, because existing varieties are not able to fight against the disease and pests attacking on the crop. And if you are not using agrochemicals to protect them, then also you will not getting the optimum yield from the crop. So, as traditional breeding techniques failed to keep pace with demand and to provide sufficiently fast and efficient systems for crop improvement, another technology called tissue culture got developed. What does tissue culture means? It was learned by scientists during 1950s that whole plant could be regenerated from the ex plant, and any part of the plant taken out and grown in test tube under sterile condition in special nutrient medium. Now, what is this thing? We have studied the traditional breeding techniques, the hybridization technique, which in which we are hybridizing or fertilizing the desired pollens and the ova of the plant in a certain conditions, in field conditions only, but that is also not too successful in getting the desired plant. So for the replacement of that thing, we have started with a tissue culture. Tissue culture is simply nothing. What you are doing in that, you are taking the tissue, especially the meristematic tissue. Meristematic tissue is what? Meristematic tissue is the tissue of the new growing plant regions, and you are taking this tissue from the above ground part of the plant. Means generally we take the new leaves for taking these tissues. So these new leaves are explored for gathering this tissue. Now this tissue is cultured in the artificial media. Now it is said that if you are taking a tissue which has the potential to generate or develop a new plant itself, that type of tissue is only taken. Okay, and that capacity to generate the whole new plant or whole new organism is known as totipotency. So totipotency is the potential of a cell or this tissue that can make or develop a new whole plant from a old tissue or old plant. So in 1950s this technology has been used for generating new plant from the older plant, from the ex plant, and that part or that work is done in the test tubes. Means basically this has been done in a laboratory in test tubes in a sterile conditions. The medias are taken with a good amount of nutrients which are required for the growth of the tissues. Now the tissues are cultured so many times that can able to produce a new plant without, like if you are growing a plant you need a seed or any vegetative part to develop into a new plant, but here you are taking only a cell or a tissue to convert that tissue or cell into a new plant. And the new thing in that is you are uh, taking that particular plant tissues which are having a specified or desired characters which you want in your plant, in which the plant you want to grow should have those desired characters in that plant. Okay. So with the help of the property of totipotency, we are started with tissue culture. Now what happens? It is important to stress here that the nutrient medium must provide, a nutrient medium would have two things mainly: the sucrose. Sucrose will help in providing the carbon to the multiplication of the cells, and the other things are inorganic salts, vitamins, amino acids, growth regulators for the multiplication. That must be there in the nutrient media. Now, by application of this method, it is possible to achieve propagation of large number of plants in a very short duration, which is known as micro propagation. So, in tissue culture, you will have first property of the that tissue or the cell of totipotency, means that cell should have the ability to generate a whole plant from the cell or the X plant. And with the help of this technique we can do propagate large number of plants in a very short duration, which is known as micro propagation. Now these plants will be genetically identical. Of course there would be no kind of variation in this thing. You have studied also in the types of reproductions, that asexual and the sexual ones. So asexual, it's a kind of a asexual reproduction, because, or you have studied the vegetative propagation. So asexual reproduction or vegetative propagations are what? They are just producing the clones of their cells. So in tissue culture also we are producing the same type of plants without any kind of genetic variation. They are genetically identical, as same as its parent plant. That is known as soma clones. Now what is soma clones? Soma clones are production of the genetically identical plants from the X plant, from the X plant. There is no kind of genetic variation in it. Its application has been seen also in production of various plants like tomato, banana, apple in a commercial level. Now, how this thing happens in laboratory, how tissue culture be performed in laboratory, for that we have some certain steps. First, another important application of method is recovery of healthy plants from diseased plant. Even if the plant is infected with virus, the meristem is free of virus. As I told you, meristem is the new growing cells. Meristematic cells are newly growing cells. They are present either in apical or axillary part of the plant. So you can recover a plant, diseased plant, from healthy plant with the help of growing the meristematic tissues of that plant in a test tube or in the laboratory condition. One can remove meristem and grow in vitro. In vitro is means in lab, obtaining a virus free plants. Scientists have succeeded in doing so in the case of banana and sugarcane and potato plants. There are many plants which has been explored with this technology. Scientists have isolated single cells from plant, and after digesting their cell wall have been able to isolate naked protoplast surrounded by plasma membrane. If you remove the cell wall, so this cell wall has been dissolved, the rest content is containing only plasma membrane and the micro cell organelles. Isolated, that is known as protoplast. Isolated protoplast from two different varieties of plant, each having desirable characters, can be fused to get a hybrid protoplast. So what you are doing, you are taking two different cells, removing their cell walls, and you are getting the protoplast of having two different types of desired characters in both the species, and you are fusing them at a in a laboratory. That is known as somatic hybrids. You are making somatic hybrids. There would be no variation in this because you are just combining the characters of two different species without any kind of exposure to UV light or any kind of mutagens. This is known as somatic hybridization. This process is also known as somatic hybridization. So, uh, imagine a situation when a protoplast of tomato is fused with that of potato to make a pomato, a type of plant which is having production of tomato and potato on the same plant. So that is also has been done with the help of this process, somatic hybridization, and it is also a technology inside tissue culture only. So tissue culture has various applications. One of them is somatic hybridization, where you are fusing the desired characters plant tissues in a in vitro conditions. So this is what resulting into pomato. Unfortunately, this plant did not have all desired combinations of characteristics for its commercialized utilization. So that's it for today. Thank you.